The overall goal of this experiment is to provide a simple and clean procedure to form 4-phenylquinazoline from the reaction between 2-aminobenzophenone and thiourea in dimethyl sulfoxide.
This method can help answer key questions in the chemistry field, such as mini-scale preparation of chemical compounds and the associated purification techniques. The main advantage of this technique are that the reaction is very clean, as demonstrated by gas chromatography mass spectroscopy or GC mass analysis, and the product can be easily purified by means of preparative thin layer chromatography or TLC.
To prepare the reaction mixture, add a compatible magnetic stir bar to a 2 to 5 milliliter microwave reaction tube. Use an analytical balance and weigh 0.0866 grams of 2-aminobenzophenone and 0.0988 grams of thiourea into the reaction tube. Then, transfer 5 milliliters of DMSO to the reaction tube. Seal the reaction tube with a compatible aluminum cap containing a rubber septum inlet. Vigorously shake the tube on a vortexer for 1 to 2 minutes to dissolve the reactants. Next, turn on the microwave reactor and put the microwave reaction tube in one of the 8 tube holders. Set up the reaction parameters through the display screen on the microwave reactor, such as the location of tube, the type of tube, the reaction temperature, pre-stirring duration, microwave absorption level, stirring speed, and reaction time. Once all